Welcome. We are the Universidad Nacional Autónoma de Honduras student chapter, and we're going to present to you a look at the coronavirus through bioinformatics. My name is Jose Ramon Cartagena,
My name is Andrés Roberto Romero.
And my name is Sofia Melgar.
These are our discussion points, objectives, introduction, materials, procedure, analysis, conclusions, and references. So to start, our objective is to spread the use of bioinformatics as an open source tool for topics of global importance, such as the knowledge about segments of the coronavirus structure. Have you ever wondered how researchers work to analyze the COVID-19 virus? It is surprising to imagine everything that an engineer, a doctor, or a biologist do on this topic. But what do you think if I tell you that there are tools that you can use to learn as a researcher about the coronavirus from home and with the use of your computer and an internet connection? All these techniques and tools are encompassed in bioinformatics, which is a discipline where biological data and a computer or computational techniques help to store, organize, and analyze that data. Behind bioinformatics, they are genetic engineers, chemical engineers, computer engineers, biological engineer, chemists, biologists, and so on. Would you like to be one of them? I will explain a little to you about this topic, but first, well, I have to talk to you about the coronavirus in order to begin our experiment with it. To introduce ourselves to the topic, we need important concepts. These concepts are bioinformatics, amino acids, protein, and glycoprotein. So, bioinformatics is a discipline where computational or informatics techniques help store, organize, and analyze biological data. Now, an amino acid is a carboxylic acid with an amino group in its alpha carbon. The union of several amino acids make up a protein, and each protein is a different depending on its amino acid sequence. Now, a glycoprotein is a different, let's say, type of protein. This one contains proteins and sugar in its sequencing. These sugars are saccharides, or more simply known as carbohydrates. Now, let's talk about the coronavirus. This virus, like most viruses, is made up of generic material it's made up of genetic material in its interior, RNA in this case. Also, it's made up of a membrane made up of proteins and lipids and spikes. These spikes are made up of the glycoprotein we talked earlier. And it's really important because this one is called glycoprotein or antigen, which are the virus's antigens.
So what can we do to analyze different coronaviruses? We can use online databases to retrieve necessary information and compare between different organisms, coronaviruses, and their structures and similarities. This is possible using online bioinformatics tools. In this case, we chose UniProt and Blast, which is basic local alignment search tool. And we chose to work with the following species: canines, such as dogs or wolves; felines, such as cats and tigers; equine, such as horses and zebras; bovine, such as cows and buffaloes; and last but not least, bats. What do we need to do it? So we just need a computer with a word processing software, such as Microsoft Word, and internet connection. Now, the important part, how will we do it? Using UniProt, our online free database of choice, we can retrieve information on the glycoproteins, such as organisms, coronaviruses, taking it to the Blast tool to compare them. Blast is actually extremely helpful. We will be learning how it uses an informatic algorithm that compares as much proteins as we want to compare. It can do a query cover, which is an overall an overall coverage, and it compares its structures. Also, we can make a percentage identity, which is a percentage of how much amino acids are identical in the sequence or following of the same sequence. And also a phylogenetic tree, which is a descendants of organisms and homology between them. Just like a family tree, I'm pretty sure we're all familiarized with, like the ones we made in preschool or elementary school to get to know our descendants. Basically, using the UniProt as our online database and Blast as our main tool, we can contrast SARS-CoV-2, which is our human coronavirus, the one we've been hearing most of the pandemic, to other organisms' coronavirus, like we said before, for example, like cats or cows. And as I said, last but not least, a bat coronavirus, and see its similarities, descendants, and its structures. But let's get our hands on our analysis.
Okay, so how are we going to do this analysis and how are we going to use a UniProt and the Blast? Okay, so first of all, we're going to head to the UniProt database. Here, what we're going to do is that we're going to look for the information of the SARS-CoV-2 or the COVID-19 and the other coronaviruses of the other animals. So first of all, we're going to go to the search bar and we're going to type in the code for the SARS-CoV-2 glycoprotein S. So it is, as you can see it here, P0DTC2. Once you touch the enter key, you will be redirected to the page where you can find all of the information about the coronavirus COVID-19. So you can find information about the protein, the gene, the organism, and the functions. First of all, I want you to look at this page and want you to analyze and read what information can you collect from this main page. And that way you can identify, okay, I'm searching, for example, the COVID-19, like the example we have right now. You'll have to analyze this page and say, okay, I am in the page that is giving me the information for the SARS-CoV-2. Once you've done that, you're going to go to this little menu bar on the left, and you're going to click the structure tab. Now, you can see we have a lot of structures for the SARS-CoV-2. The one we are looking for is the 7C2L. We're going to search it here, 7C2L. And as you can see here, it will generate the structure for the COVID-19 as we know it. So this is the S-glycoprotein structure of the COVID-19. So you can analyze, this is this is found in the outermost part of the virus, and it's a quite well representation of how we can see the coronavirus, and we're going to see it along the other organisms coronaviruses. So after we have seen this structure and analyzed it a little bit, and you can tell it's the same structure that you can find in the outermost part of the COVID-19, we're going to come to the left menu, and we're going to go to the sequence tab, where we can find the sequencing of amino acids in the glycoprotein S of the COVID-19. This code, well, it just gives us the order in which the amino acids are organized to form the glycoprotein. What we're going to do here is that we're going to look for this button right here that says FASTA, and we're going to click it. And it will redirect us to this page, which contains the FASTA code. So the FASTA code is just a format used in bioinformatics so we can sequence the proteins. And now what we're going to do next is that we're going to open our word processing software. Okay, right now we are in the interface of Microsoft Word, which is the word processor that we commonly use. So what we had to do was to copy the FASTA sequence that we had generated on the UniProt and paste it here. So what we're going to do is that we're going to paste it, and you will see your sequence right here. And this is the same procedure that we're going to be making for all the other coronaviruses that we've selected, mentioned by Sophia at the beginning: the canine, the feline, the equine, the bovine, and the bat. You can find all of these codes in the handouts that we are going to be given to you. Now, as you can see, we have collected all of the FAST codes for all of the coronaviruses that we want to compare. So the first one right here is the COVID-19, and the other five that are presenting it are the FAST codes of the other five organisms coronavirus. So what we're going to do now is that we're going to take this to the blast. Okay, so what we're going to do is after repeating the whole procedure once again for all of the other organisms coronavirus, we're going to collect all of their information in a document. As you can see, the first one is the code for the fastest sequence of the COVID-19, and the other five are the ones for the other organisms coronavirus. So we're going to head to the blast, as you can see it here, and you can find in this page this section called Webblast, where we are going to go to the protein blast, which will help us to align the sequences. So first things first, we're going to see here a box that says enter query sequence. What we're going to do there is that we're going to copy the COVID-19 sequence and we're going to paste it. Only the COVID-19 sequence, the first one that we collected, the human, because this is the one that we're going to be comparing to the other five sequences. And next, we're going to take all of the other sequences, we're going to copy them, and then we're going to go back to the blast. And before pasting them, we have to select this box here that says align two or more sequences. And this will allow us to run the blast so it can align, and then it will compare all of these sequences. So the next step, we're only going to paste the other sequences. And when I say copy paste, I mean everything, not only the FASTA code, but also this title that appears over here, because that will help us identify which coronavirus we are talking about. Once we have done this, we're going to go scroll down and we're going to find this button that says blast. So we're going to click it, and then the blast will start processing all of this FASTA sequences, and will give you a table of results where we can clearly see um, the results that our colleague Sophia was talking about earlier, which are the query cover and the percent identity. So as we can see right here, the one that has the higher query cover is a bat coronavirus, which is of 100%, and also a percent identity of 97.41%. So we can say that the bat coronavirus is the one that most resembles the human coronavirus. I mean, it's not a surprise for all of us, but we can prove it with all of the results that we are seeing right here. And you may say, well, what if I want to compare, and what if I want to see which other coronaviruses resemble each other? You have this option called distance tree of results, where you have the comparison between the other types of coronavirus between themselves and which ones align with which others. So first of all, we have right here at the bottom the SARS-CoV-2, the COVID-19, and we already know that it has a match in the bad coronavirus. But we can also see that we have other two families. First of all, we have the um, spike protein for the feline and the one for the canine coronavirus. So we can see that these two resemble each other, and also do the bovine and the equine. So we can say as a conclusion that bioinformatic tools are really, really important. This is just one of the uses that we can give to these tools. Um, and you can broaden all of your knowledge about this, and you can start investigating, and you can start using these tools that we have provided you with. They're free to use, and we hope you can get the most out of them. Thank you so much. And now to finish everything off, we have to, of course, give credit to all of the places that we took information from. Our open source tools were the UniProt database and the basic local alignment search tool, also known as Blast. And also we have cited articles and books. We have cited the alignment of SARS-CoV-2 in comparison with other coronaviruses, and also the biochemistry by Matthews, and biochemistry, the molecular basis of life by J.R. McKee. That will be it from us. Thank you so much for your attention, and we hope you can take a lot of things from our project.